So, what, do you guys think it's like really hot here?
It's
Like
humid.
really?
It's muggy.
This is ridiculous. So I obviously I graded y'all's live exams. For the most part, like cumulatively, I did really good. There were 5 perfect scores on the exam, which is like I've never seen that before. So that was pretty good. However, again, if you didn't do how you wanted, we talked about this last exam, and I'll keep harping to you all. My job isn't just to be here to teach you all the material. Otherwise, you would just be in the book, right? I'm supposed to explain it in a way that I understand it. But secondly, I'm trying to push you all and motivate you all to do better every time. Okay, so if you didn't do how you liked on the exam, and on the lecture exam too, this can apply to anything, it can apply to life in general. If you don't do how you want in life or in class or whatever, you change what you're doing, okay? And this was a mistake that I fell into many, many times, is that I thought my issue was that maybe I wasn't putting in enough time. No, it was the way that I was studying. I had to change what I was doing, okay? So don't ever fall into the trap of doing the same thing and expecting a different result. Nothing changes if nothing changes, okay? So, and we'll probably have this conversation again after the second exam. Hopefully everybody gets hundreds, but we'll see, which is Monday, okay? So we have that exam Monday. I will publish we'll say like a practice quiz or like a quiz of sorts on Blackboard for this weekend. I have concepts posted for the video assignment. If you haven't done the last video assignment, please get that done as soon as possible. And the second video assignment, which I'd like it by Sunday night or by the end of Monday. Understand that homework is 10% of your grade. So even if you got a perfect score on all the exams, if you didn't do the homework, you might not even get a 9. I mean, I would give you an A, but if you miss a couple points, you probably wouldn't be able to get an A in class just by not doing those video assignments. And there's, of course, a reason why I'm doing it. Okay, so we finished up talking about facilitated diffusion, and understand that when you hear the term diffusion, hold on. When you hear the term diffusion, we're talking about moving from high concentrations to low concentrations always, okay? High to low concentration. And actually, let's come back to a point. What does concentration mean? What is it? We've talked in detail about concentration. When I'm saying concentration, what am I talking about?
It's the amount of substance.
The amount of substance. The amount of particles. How do we measure the amount of particles? Through molarity and moles based on the periodic table. They're given to you right there. Those atomic weights are essentially giving you, and I don't know if maybe I mentioned it, but the atomic weight on that periodic table, you remember we calculated the atomic weight for certain molecules. Those numbers there, say for example, oxygen is equal to 15.99. And this is how we calculated atomic weight. This is how we got molarity. Molarity was equal to moles per liter. So this is telling you how many particles are in a liter per 1 mole. And we know that through Avogadro's number. However, this number is telling you the grams per mole. So if I weighed out 15.999 grams, and put it into a substance, that's 1 mole, okay? So, I'm bringing this up, obviously this is not pertinent to the exam, but it is important because we're talking about high concentration to low concentration. And I don't want you to forget when I'm saying concentration, you know how to measure that already. You know what I'm talking about because we're talking about moles. Moles, Avogadro's number, that's how many particles are in the solution. Okay? So if I said I had 2 moles out here and 1 mole in here of whatever substance, that means that whatever particles are out here are going to flow inside the cell. High concentration to low concentration. And that is the principle of diffusion. Now, the reason why the particles go into the cell or move across a membrane or whatever is different. The reason why they move is different. But it's still the concept, high concentration to low concentration. And what were the reasons? We talked about simple diffusion. These were molecules that could freely cross the membrane, meaning that they'll always move from high to low. But then we had help sometimes through facilitated diffusion or facilitated transport. This was using a protein at the surface of the cell. And for whatever reason, whether it was voltage gated, whether it was chemically gated or mechanically gated, that's what caused the protein to open, and then the solutes would flow into or out of the cell, depending on the concentration. Okay. So this is an example of a facilitated diffusion process. This one happens to be a GLUT transporter. This is not the last time that we'll see it. This is how glucose gets into some types of cells out of the blood. And there's multiple different types. We'll see GLUT 2s and 4s in this class, and you'll see them next class as well. This is introducing the concept, and it's pointing out to you that when glucose gets into the cell, we're going through we're becoming either glycogen or glycogen is breaking down to then release glucose out into the cell. Okay, not necessarily important. Here are the passive membrane transporters, which we already talked about. This is a good summary slide here. We have simple diffusion where they're moving through the membrane freely. But understand, it doesn't necessarily have to be through the membrane, right? I showed you an example of this, or I told you about an example of this jug, and I put whatever solutes I want on top of it, whether it was salt or some colored powder, and I put it on top, and those particles would diffuse down freely. That's high concentration to low concentration, and it will occur always. Okay? Then we had the carrier-mediated. Don't forget, carrier-mediated is whenever the protein so this is still under the category of facilitated diffusion. If you ever see a protein in the membrane, it's still facilitated transport. Okay, that's what that means. But with carrier-mediated, one side of the protein is closed at one point, and then the other side is open on opposite sides. And they will only exist in those two states. There's never going to be, they'll be open at the same time. It won't occur. Okay? And so it requires a ligand or a chemical solute, whatever, to bind on the inside, changes the protein structure, and it opens towards the inside or the other way around, depending on the concentration gradient. Okay? Because we're still talking about diffusion. The other one was channel. And I told you that channels, when they're open, whatever it is will flow freely through the membrane. There is no open and closed states. Okay? It either opens or it's closed. No in between. And when it's open, things will flow freely into the cell or out of the cell. And then there's something else that we're talking about, which is osmosis. This is another type of passive transport, but it's only moving water. Okay? So this one's kind of different, and I want you all to pay attention. Osmosis is the movement of water down its concentration gradient directly through a lipid bilayer, through water-specific channels called aquaporins. The aquaporins is not necessarily important, but understand this is the movement of water, not solutes. Okay? So whatever this is, you have to get out of your mind that we're not talking about concentration of solutes anymore. Well, we are to an extent, but we have a membrane that cannot we have a membrane that cannot move solutes. We are only talking about water. This is osmosis. And so I like to introduce, the equations aren't necessarily as important, but I want to introduce these terms. And actually, maybe, because I have them on a different slide too, we might move to the second slide. Yeah, let's go ahead and move to the second slideshow because I want to introduce this concept before we talk about it. So in order to understand osmosis, you first have to understand this concept of tonicity, okay? And this is similar to concentration gradients, except we're really just talking about water, okay? A mix between water and concentration. Tonicity is describing a solution relative to another, okay? Now, this is similar to concentration, but concentration is very descriptive. You say I have 1 mole, I have 2 moles, I have 3 moles of whatever concentration. Okay? Tonicity is relative to another solution. Okay? And there's 3 terms that you have to know. There's hypertonic. And so when I'm saying hypertonic, I mentioned to you that this is relative to another solution. Okay? So you always have to know that you are describing it against another solution. So hypertonic means that the concentration of solute is higher than the one that you're comparing it to. Hypertonic. Hypotonic means that the concentration of the solute is lower than the one that you're comparing it to. And we could be talking about any solute. Isotonic is when the 2 solutions are equivalent in concentration. We can be talking about anything. Okay, and the formal definition is the ability for the solution to change shape of cells by altering the cell's internal water volume, and I'll get to that explanation in a second because this is how we're describing the differences in solution concentration between cells. Okay, this is an important concept to understand because when we're talking about the cells, when we're talking about the blood, when we're talking about the space in between the cells, it's all made of water, but you understand now that we have thousands, if not millions, of chemicals existing in that space. So it's very easy to describe the concentration of a solution in general compared to another one. And it's a diverse number of chemicals. We're not talking about 1 or 2 or 3. Okay, so hypotonic, the solution that you're comparing it to is lower in solute concentration, hypertonic higher. Then isotonic is equal. Okay. One thing to understand is that water moves from hypotonic to hypertonic. Now this is opposite to what you've kind of been thinking. Solutes move from higher concentration to lower concentration. Solutes, the chemicals themselves will always move from higher concentration to lower concentration. However, if you stop the solutes from moving, don't ever forget, the solutes aren't moving. It's only water. Water will move from hypotonic to hypertonic. And it begs the question, why? Okay, so the best way to explain it is just explaining it live on the board. And so I drew it on that board, but I don't think that that's easy to see. Okay. So let's see if I can use my finger. Okay, I can use the. Okay. So let's say that we have a beaker, okay, a large beaker. And we have a membrane in here that cannot move solutes. Okay? And let's say we have some solutes here. Boop, 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 boop. We got a lot of them right here. We have concentration of water. So when I'm drawing the water here, understand that it's filling this whole beaker up to its level, okay? Then we have another solute here. And we have water concentration here. From what I just explained to you, which one of these solutions is hypertonic, which one's hypotonic? What is this? Hypertonic, exactly. Hypertonic. And this one will be hypotonic. Okay? You always have to have something to compare it to. So if I ever tell you something's hypertonic, you need to know immediately whatever the other thing that I'm talking about is has to be hypotonic. Okay? And vice versa. I don't have to tell you the concentration of both solutions because it's relative. If I tell you hypotonic, you have to know, even if I don't even mention the other solution, you know it's there and it's hypertonic. It's the opposite. And if I say isotonic, you always have to understand there's another solution. And the other solution would be equal, isotonic. Okay. So, if under what we've already explained, where do you think that the solutes would move? What we've explained before. Get osmosis out of your head. Where would the solutes move?
High to low.
From high to low. If the solutes move, they would move from high to low. That's diffusion. But osmosis, you have to have a barrier that cannot move solutes. There has to be a barrier where you cannot move solutes and only the solvent or water can move through. Okay, so if these solutes can't move, and let me go ahead and put the... If these solutes can't move, where would water have to flow? From hypotonic to hypertonic, like I already explained. But why? If the water is equal here and the solutes can't move, the only next best option to get this concentration to be as equal to this one would be to move the water, increase the water concentration so that this is more dilute. I increase the volume of this. So now, this solution becomes less hypertonic or more hypotonic. It becomes more dilute. And the water down here will decrease. Because the volume decreases, you're packing these amount of solutes in a solution that now has become smaller. So now, inherently, it becomes more concentrated. So let me draw a new line. Let's pick a new color. Let's do this light blue. So water will flow from here to here. And so now this water concentration comes down to this level, and this water concentration comes up to this level. So now what you can see is that this solution has become more dilute. And this solution has become more concentrated. And it does that until the concentrations become equal to each other. Okay? Does that make sense to everybody? But you have to have a barrier that cannot move solutes. Okay? Now I'm not saying every solute, but it has to be some. Okay? Now why is that so important? Why do you all think that that's so important to understand this process? I told you that the body's made of water, but why would this be so important to understand? How does it relate to our body? We have a lot of water. We have a lot of solutes. How does it relate to our body?
If we want to keep like more nutrients or something in one part and then move our fluids.
Okay, this is how the kidneys work. The kidneys are filtering, so you're right. If we want to keep some nutrients and the digestive system, this is how the kidneys are filtering our blood and making sure we empty out certain things, regulating blood volume. However, there's the big picture concept here. I have solutes in my body. I have all kinds. We talked about so many that are inside one cell. We're made of tons of water. Okay. This is describing the cell. This is the plasma membrane. You want to go back to the PowerPoint slide? Let's look. Yeah, this is. I've shown you this figure already. Our plasma membrane is this barrier. We have a barrier that cannot move certain solutes. So we're creating this hypertonic and hypotonic solution in our cells. And what's bathing it is all this water, all this blood. So we have to make sure that we maintain and, understand this process, but we do it because we have to understand that if we add a lot of solutes to the blood, or if we take away a lot of solutes from the blood, that will affect where water's moving. That will affect the size of cells, and then thus the functioning of the cells. The reason why that's so important is because this is the plasma membrane. It's a barrier that's permeable to water, that's impermeable to other solutes. You remember I told you it doesn't have to be all the solutes, but it has to be some that create this hypertonic, hypotonic. Okay? So that's osmosis. Osmosis, there's rules. You have to have a water-permeable membrane that's impermeable to solutes. Not all of them, but some. And water will flow from hypotonic to hypertonic. Now, one of the most prominent cells, well, fake cells, that are floating through a solution are the red blood cells. And you can see this happen live. Okay? So if you create a solution and you make the solution isotonic, and you put red blood cells in them, they will maintain normal size because water is not flowing in and out in opposite. Water is still flowing in and out because it's permeable. However, it's flowing out at the same rate that it's flowing in, so the cell maintains its size. However, if you have a hypertonic solution and you put blood cells in, the hypertonic solution is like, for example, this jug of water. The red blood cells is the other solution that we're comparing it to. So it has to be what? Hypertonic solution. Yeah, so it has to be what? If I say that the solution that we're putting in is hypertonic, you always have to understand there's another solution. And the other solution on the opposite side of the membrane would be what?
Hypotonic.
Hypotonic. So if you put that inside the blood cell, water will flow from hypotonic to hypertonic, and the cell will shrink. That process there is a term for shrink. Well, you can just say shrink. That's okay. The cell will shrink or shrivel. Water will flow out of the cell. You can literally see this happen live. And the same thing with hypotonic. If the solution that you're putting in is hypotonic, you know that there's an opposite solution. That opposite solution is inside the cell and it's hypertonic. So the hypotonic solution will flow into the cell and it will burst or cause a term called lysis. And I would like you to know that term. Lyse, L-Y-C-S-E, or lysis. I'll use that term throughout the rest of this class. It's the bursting of whatever you're talking about. The cell bursts. Okay. So this is very important to understand osmosis. Okay, so that's tonicity. And so, yeah, I sort of already explained that osmolarity is concentration within water. However, I do want you to know that throughout this class and now from now on forward on exams, I will use this nomenclature, these two brackets to describe concentration. If you put anything in two brackets, and of course you'll see in your textbook, like if I say sodium, You all should know that symbol now. And I put brackets around it. We're talking about concentration. The brackets mean concentration. So I could say this, boom. Another symbol you should know. And what am I saying when I write that?
The concentration of sodium is greater than the concentration of potassium.
The concentration of sodium is greater than the concentration of potassium, correct. So, this is the nomenclature I'll be using from now on. You all need to get this term because it's, it's a lot. Writing concentration a lot is not fun, so I'm not going to do it anymore. Okay, here is like sort of a summary slide of what can move through the membrane. Again, gases, nonpolar or hydrophobic molecules can move through the membrane. And when I'm saying nonpolar, you all know what this directly relates to. It relates to the covalent bond, nonpolar, okay, which is also considered hydrophobic. Then you have small polar compounds like water. And typically, large compounds cannot pass through. For the most part, large molecules cannot pass through the membrane. There's another term called lipid solubility. And I don't know if I mentioned it yesterday, but these are molecules that are soluble in lipids. And so they'll be able to pass through, the membrane freely. They're lipid soluble. And also, the smaller the compound, the higher likelihood it has a chance of crossing the membrane. Okay. Now, this is another slide explaining carrier-mediated versus channel-mediated. I'm not going to explain that again. However, there is an important point that I want to get across to you all, is that why do we need these channels and carriers? Why do we need them?
To get things across the membrane.
To get things that cannot pass. To get things across the membrane that cannot pass inherently. Hopefully you all understand that. Always come back to what in the world are we talking about? Okay? Tonicity. Usual summary slide. Okay. So now, let's talk about active transport. We've talked about passive transport. This does not require energy. No energy required. The energy is the concentration gradient. Like the phony energy. If you have to think of it as energy, it's the concentration gradient for passive diffusion. Now, there's another type of diffusion, or not diffusion, another type of transport called active transport. And this requires energy, oftentimes in the form of ATP, but not always. It's not always ATP, but oftentimes we're talking about ATP. It could be something else that's releasing energy, and oftentimes in the form of ATP. Okay, question. So we know that we need transporters for molecules that are incapable of passing through the membrane. Why would we need energy to move molecules across the membrane? I want somebody who hasn't spoken to sort of think about this and say something. Why in the world would we need energy to move a molecule across the membrane outside of the fact that it can't move on its own? Well, okay, I'll give it to you, but it's because active transport is moving molecules that don't want to move from higher concentration to lower concentration. We're moving them in the opposite direction. Remember we talked about spontaneous reactions? And we said they'll go on inherently. Well, that's what high concentration or low concentration is. That's something that's very spontaneous. It doesn't require energy to go on. It will go on inherently. However, what if you want to bring a molecule from low concentration to higher concentration? We have to put energy into the system to allow it to happen. We cannot, it does not occur on its own. If I take the salt, again with the example of the salt or the colored powder, and I put it in this water, there is no possible way that you would be able to take those particles and move them back up to the top where I put them. There's no possible way unless you did something like added heat, maybe I boiled the water off, that's energy. Or if I took it to a chemical lab and maybe added like some sort of way to pull the molecules up. The concept is though, it will never go back unless you put energy into the system. Same with salt water. Everybody knows that if you take water out of the ocean and you boil it, you'll get the solutes back. It'll turn into salt, right? That's energy going into the system. That's moving the particles back to where they were. You cannot move particles from lower concentration to higher concentration without energy into the system. Okay? And that's what active transport is trying to do. So if I ever tell you active transport, you need to know, one, it could be that a molecule can't cross on its own. It might be super large, like in the case of glucose. However, oftentimes we are moving a solute from low concentration to higher concentration, the opposite. That's why we need active transport, and it requires energy to do this. So, there's multiple different types of active transport, which we'll talk about. So again, this is the biggest concept for active transport, moving particles, and when I'm saying against a concentration gradient, means low to high, okay? Oh, I didn't know I had these on these slides. So if I'm looking at these two figures, because understand, when we're moving through, whenever I showed you that facilitated transport, those proteins, those are still chemical reactions that are occurring. You just don't see them. Okay, we're not worried about the chemical reaction. However, we still have to think about it a little bit. If I'm looking at active transport, what type of reaction would that be? Somebody tell me who hasn't spoken. What type of transport would that be? Or what type of reaction? Endothermic or exothermic? Be endothermic. We're putting energy into the system, okay? These are still reactions that are going on. Even the transporters that are moving the solutes, they are chemical reactions, and I just told you that you require energy. That was the definition of an endothermic reaction. We had to put energy into the system to get to the other side. Exothermic was something that we were getting energy from. Okay, like the process of ATP breaking, and I think we mentioned that. Hopefully we did. But endothermic, we're putting energy into the system for the reaction to happen. Exothermic, we're getting energy back to use it for something. Okay. So in the case of active transport, these would be endothermic reactions. We're using the energy to do something. Okay, so this is where we're talking about ATP. I've mentioned this molecule ATP so many times. Okay, and here it is. This is the structure of ATP, and I don't know if I mentioned to you all last exam, but I do want you to be familiar with the structure. Okay, you don't have to memorize the structure, but I want you to be familiar with it. What do you notice here? Somebody who hasn't, I want you all to get out of your comfort zone. Don't sit in this class and be comfortable. Remember what we talked about last exam? This is not about being comfortable. This is about learning and being a little bit uncomfortable. What do you all recognize about this molecule right here? Who said that? It's a ribose sugar. How do you know? Because it has an oxygen right there. Exactly. What do you all notice about this? What does that look like to you? You don't have to tell me the exact thing, but what does that look like? Okay, it's a purine. It's a nucleic acid. That's a nucleic acid that happens to be adenine. ATP stands for Stands for adenosine triphosphate. This is what ATP is. So you have a nucleic acid, adenosine right here. It's a ribose connected to adenine. Adenosine. Tri-3-phosphates. And you remember I told you oxygen almost always is bound to phosphorus. Almost always. You will not see these phosphates. And you remember the chemical formula I showed you for that? It was PO4 minus. So for every single phosphate that you're seeing here, you can count 1, 2, 3, 4 oxygens. Now, obviously, they're sharing them at some points, but a phosphate is PO4 negative. Now, you see 3 phosphates here. You got 1, 2, 3, and you got all these oxygens. Well, a lot of these oxygens have negative charges, okay? And understand that this is the molecule that we're making when I'm talking about energy. And I've told you this time and time again, when we're breathing oxygen, this is the purpose, this is what we're making. When we're taking in glucose, when we're taking in all these carbohydrates, when we're taking in these lipids for energy production, this is what we're making. And we're making it in the mitochondria. These molecules are incredibly important, and I'll explain why. You have these 3 phosphates here connected to this molecule. Well, what do you know about like charges? What do you know about negatives and negatives? They repel each other. Positives and negatives, opposites attract, negatives and negatives, they repel. Everybody should know that. Well, what do you notice here? We got a lot of negative charges bound together. These phosphates are bound together and they're holding oxygen and they got all these negative charges. Don't you think that there's a little bit of tension pushing between these 2 molecules? There's a lot. These molecules, these phosphates do not want to be bound together. And actually, the more that you add, the more tension that you're developing. And what do you know about what these bonds are? They're holding the electrons there. What did I tell you energy was? What did I tell you the definition of energy was? The movement of charged particles. And I told you that there was something called the chemical bond, which was holding the potential energy. Potential energy becomes kinetic energy. Breaking a bond releases a lot of kinetic energy because we're releasing charged particles. Well, for some reason, and some chemists can explain it to you, this molecule is very unique in that we can add 3 phosphates together and there's a lot of tension between these oxygens all over. A lot of tension. But for some reason the molecule holds. But it's literally like a grenade. These bonds are ready to break. Okay? So, when you add these phosphates, you have this molecule that for some reason will exist on its own. Now, I can tell you that it required energy to make these bonds. It required a lot of energy to push them together, almost like 2 magnets on the opposite side. You push them together and for some reason they're sticking together. That required energy. That's why we needed glucose. That's why we need oxygen. That's why we need all those carbohydrates and lipids. Because it took a lot of energy to put this together. But it's literally ready to snap. And whenever it does, you chop this bond between these 2 phosphates and energy goes everywhere. And you can put energy into the endothermic reaction. Now, breaking that bond would be what type of reaction? Breaking this bond would be okay, it would be a hydrolysis reaction, but it would also be incredibly exothermic because we're releasing lots of energy by breaking that bond. This is why we need ATP, and this is what we're using in active transport. But I do, listen, what I just explained to you about ATP, I want you to know. This is a very important concept. Don't ever forget what this molecule is actually doing. This will certainly be on the exam. Now, when we're talking about active transport, okay, I brought up ATP because ATP is required, energy is required. This is what energy is coming from. There are 2 types of active transporters. There's primary active transport and there's secondary active transport. And then there's other ones, but there are 2 main types, primary and active. The difference between the 2 is that one of them is directly using ATP. for its transport. The second type is indirectly using ATP. Okay? So primary active transport is directly using the breakage of this bond to then allow for the transport. Secondary active is indirectly using it, and I'll explain them in detail in a second. So primary active transport. We have this one transport, and I wish I had a bigger figure. Well, okay, primary active transport requiring energy. Well, there is, and also the concept that you just brought up, Sophie, hydrolysis of ATP is the same thing as saying using energy, because that's exactly what we're doing. We're hydrolyzing that phosphate bond. So if I say hydrolysis of ATP, I'm saying we are releasing energy into the system, okay? So I could say primary active transport and have an answer choice saying requires a hydrolysis of the ATP instead of saying requiring energy, okay? Now, one of the most well-studied primary active transporters happens to be something called the sodium-potassium pump, sometimes abbreviated as Na/K pump. This is why I wanted you to know the element symbols for the elements, because we don't write them out anymore. Na/K pump, or some people do NKA for active transporters. Some people say sodium-potassium ATPase, being the ATP enzyme. However, this is the most important transporter in your body. It's the most abundant transporter within your body. And so a lot of people have spent their lives studying each step in this process. So we're going to know. Okay. The first step is that it's, so this is it. So the yellow side is inside the cell. The blue side is outside the cell. Okay, and you all can see the plasma membrane. And this is, what type of, well, this is a carrier. Hopefully you all can identify it. It's open on one side and closed on the other at the same time. However, this is active transport. This is not carrier-mediated facilitated diffusion. Okay. So, first thing that occurs is that 2 or 3 sodiums from the inside of the cell bind to the inside of the transporter. Okay? That binding, along with ATP binding, allows for this channel to then close. Now, the closing of the channel is stimulated by the hydrolysis of ATP, and you can see that right here. And hopefully, if you have it on your screen, hopefully you can see it in a little bit more detail, the words that are coming from your textbook. However, the hydrolysis of ATP, and what do you see here? You see one phosphate that's bound to the inside of the cell, on the inside of the carrier, and then you see this molecule called ADP released. Okay, what that is the same thing, adenosine triphosphate, except we now have diphosphate. So that's telling you what? You have 2 phosphates bound to that molecule. Okay, so where's the other one? It is on the inside of the carrier, or on the inside of the cell bound to the carrier. In essence, that hydrolysis reaction occurs and then releases the rest of ATP, and that phosphate is now bound to the carrier. Okay, so now the carrier opens to the outside of the cell and releases sodium. And at the same time, 2 potassiums will bind on the inside of the carrier. Then it will close and open to the inside, potassium will release, and then ATP will bind again, sodium will bind to the inside of the carrier, and then this cycle continues and continues and continues. Now understand that these things have been working since you were born. So don't think, if I give you a step-by-step process, which I will on an exam, I will have you list out each step. Now I might give you a word bank, but you will have to know this in detail. Understand that you could start at any point. Okay, because this is a cycle that's been continuously occurring, and this is the same thing through what you'll see throughout this class, there really is no starting point, because it's always been going, right? So we just give you a starting point so it's simpler to understand, but understand I could start right here, right at step 3, and you all should be able to figure out the rest of what's going on. Okay. So 3 sodiums move out of the cell, 2 potassiums move in. Now this introduces another question. Now I want somebody else who hasn't spoken to talk about this. Tell me the concentration gradients of each solute inside or outside the cell. Because you can, we've talked about it already. What will be the concentration of sodium? What would be the concentration of sodium inside the cell? We're talking about relative to the outside. Okay. Diffusion moves from what concentration to what? High to low. High to low. Active transport, I told you, moves from what? Low to high. So like I told you already, I mentioned this with diffusion. If I say something is moving via diffusion into the cell, you can tell me the concentration of that single solute inside the cell and outside. Relative, of course, you can't tell me the direct concentration or the exact concentration, but you can tell me the relative concentration. When I say active transport, if sodium is moving outside the cell, it's moving from low to high. So you know immediately inside the cell, sodium is incredibly low. And sodium concentration outside is high. Because we're putting energy into the system to move it against this concentration gradient. Now if it was the opposite, if sodium was high inside the cell and low outside the cell, we wouldn't need a carrier. We wouldn't need active transport because we could just open the channel and it would go. But because we have to put energy into the system, we're moving against our concentration gradient always. So if I say sodium's moving outside the cell, sodium concentration is low inside and high outside. We're moving against the concentration gradient. And the same thing for potassium. If potassium is moving inside the cell, you know that its concentration is very high inside the cell because it's moving against its concentration gradient. Okay, and this, I need you to know all these steps. And I'm trying to get you to think. I repeat myself over and over again because I want to get you all to think about what we're talking about. Now, so everybody understands how you can determine concentration based on if I tell you diffusion or active transport or whatever, right? Okay. Because it's very important to understand secondary active transport. Secondary active transport, the first thing I want you to understand is this is not using ATP directly. Before we were using ATP directly. The ATP was binding directly to the carrier. That's primary active transport. Secondary, you need 2 things. One, you need to understand that ATP is not directly being used, and you have to have 2 protein carriers. 2 of them. Now, it could be a carrier, it could be a channel, but the concept is you need 2 proteins at the membrane. Okay? 2 of them. Now, one of those carriers, so when I'm saying 2, it's a similar concept to the hydrogen bond. You remember, in order for the hydrogen bond to form, you had to have a polar covalent bond. You all remember that? It's the same concept. In order for secondary active transport, you have to have ATP being used with one carrier directly, and then the next one is not using ATP directly. Okay. So we just talked about this. Primary active transport is moving particles against their concentration gradient. Okay? And so, oftentimes, inside the cell, this will change the concentration gradient. So we can change concentration gradients of solutes based on active transport. Now, the change in the gradient then allows that solute, and I know this is, I'm not going to lie to you, this is a very difficult concept to understand. So I'm going to show you all multiple figures on this. However, that change in concentration gradient drives that solute back into the cell. So in essence, what I'm saying is that, say you have, sodium we'll put a membrane, and we'll say we have low sodium solute inside the cell, and we have high out here. We're essentially moving through ATP, sodium outside the cell. Okay? This is using ATP directly. This is primary active transport. We make sodium's concentration even higher. That generates a higher concentration gradient, and then sodium will want to come back into the cell. But there's special transporters that will utilize this high concentration, and it will tag along a partner oftentimes that's moving against its concentration gradient. But the energy that was formed here, the higher concentration, is so strong that that new transporter will allow sodium to come in, but it also partners along with some other molecule that might be low out here and high in here. Okay, I know that might not make sense to everybody. It's not an easy thing to understand. However, you can have two different types. You can have symporters, and this applies to all channels too. I don't want you to think that this only applies to this. When you have a symporter, you have two molecules that are moving in the same direction. What I just described to you, sodium's tagging along with another partner to move into the cell. Now, antiporter are two molecules that are moving in opposite directions through the same carrier. Okay, so in this case, if sodium was moving inside, the other partner molecule would be moving outside. Okay. Now, here's a better figure to help understand this. Is that here's a sodium-potassium pump. We're making sodium concentration very high outside the cell because it's already high out here and it's low down here, and we're moving against the concentration gradient. So we're just adding to this high concentration out here. Well, you have specialized channels, okay, specialized carriers really, that will move sodium and a partner. In this case, it happens to be glucose, which is a very common type of transporter, and it won't be the first time you'll see it. It will utilize this high concentration to move sodium inside the cell, but we'll say that glucose is low outside the cell and high inside. You all know inherently to move glucose, you can just look at glucose. This would require energy to get it back into the cell, right? Because it's low out here, it's high in here. Well, we're using the energy of sodium moving outside the cell so that we generate a very high concentration out here and a very low concentration out here so that sodium will move through and tag along glucose. Okay, it's basically like a buddy system. You have to have two transporters. Okay, two transporters. One of them's using ATP directly to cause this concentration gradient to be even higher. Then you have another carrier that uses that very high concentration to tag along a partner to move into the cell. And that partner oftentimes is low outside and high inside. Okay? Because that would inherently need energy. But it's using energy from a previous transporter. That's why it's called secondary active transporter. And this is one of the most common. This is certainly, these are called SGLT1 and SGLT2 pumps, and heavily prevalent in the kidneys. This is one of the most, a cause of many, many diseases, SGLT1 and SGLT2. Okay, here's another figure explaining it. You're using primary active transport, generating this concentration gradient to tag along the partner. Now, understand that if they're moving in the same direction, it's antiport. Now, it does happen where the second transporter moves in the opposite direction. This is also very common, is that sodium might move in and something like hydrogen might move out. Okay? This is also common. Glucose can move out. It's still secondary active transport. Okay, you all take a 10-minute break, come back at 10:07. 10:08, 10:07. So I mentioned to you that there was primary active transport, and there was secondary active transport. That had to do with channels or transporters, like protein transporters, okay? We also have other types of transport into and out of the cell. Okay? So I want you to think of what's called vesicular transport. It still requires energy, but these are not protein channels in the membrane. Okay, and you all can imagine right now that this line here is the plasma membrane. You can see the individual phospholipids. Hopefully, that's what you're imagining when you see this. Well, sometimes the membrane can create those little tiny mini cells that we talked about, those vesicles. Okay, and this is how we're transporting things in and out of cells. This still requires energy. So still think of this as transport, like the channels in the membrane, or the proteins in the membrane. The proteins in the membrane, so to speak, are these little tiny vesicles. Okay? So, vesicles, we've described them multiple times. These are smaller portions of the membrane that pinch off and create these mini cells and hold carbon. Now, oftentimes this is how we're moving large particles. Okay, very large ones, like hormones. Okay. And so vesicular transport is the movement of these vesicles. Now there's two big types. There's one called transcytosis, where we're moving between cells. So I'm taking these particles and trying to get it to another cell. And then there's something called vesicular trafficking, which is the movement of the vesicles within the cell. And do you all remember how these vesicles were moving in the cell? We said there was this transport mechanism in the cell, and we mentioned it with organelles. It was like the interstate. It wasn't necessarily an organelle. It wasn't membrane bound. The cytoskeleton, and that was protein. It didn't have membrane associated with it. And this is how vesicles, I mentioned to you, those microtubules, is how we're moving these vesicles in through the cell. So, yeah, this is very plain. But understand, we have that cytoskeleton, that's one holding the cell structure, but then also it's extravagant throughout here. Then these vesicles are moving on them. Okay, they're not just floating freely. Now, there's two really big categories. So I talked about broad types of movement between cells and inside the cell, but there's two big categories. There's a process called endocytosis, which is this, where we're taking components from the outside and bringing it in. Very, well, I guess I can't explain it. But then there's exocytosis, where whatever we have inside, we're trying to get it out. Well, I guess this is what this process is. We're trying to get these particles, we package them. This could have been a protein that was made by the endoplasmic reticulum, and you remember we said that we package them into vesicles and we move them along sometimes to a different location, and sometimes that's outside the cell. That would be exocytosis. Okay, and there's steps associated with each, and I want you to know these steps. Okay, and I have them written. So this is the process of endocytosis. Now, what I'm explaining to you is very general steps. With everything that we're talking about, though, there's individual details within here that we'll be able to apply to those specific situations. And I'm bringing that up now because we will see those specific details in this class. This is general endocytosis steps. So the first step in endocytosis, remember, we're bringing something inside the cell. So something's outside here that we need to bring in. It could be sodium, it could be potassium, it could be calcium, whatever. It could be anything. Comes to the top of the cell. It sort of hovers around an area of the membrane. And what happens is that membrane, the plasma membrane, will sort of what's called invaginate a little bit. It will come down, form a little bit of like a pit. Now this is much larger than what it originally starts as. It starts as much smaller. It sort of comes down a little bit, sort of almost catches whatever the particles are. And then you form what's called a protein coat or a coated pit. That's what this is. So now, whenever it sort of invaginates a little bit, you send a signal on the inside of the cell. And I'm telling you, there's a lot of details associated with this, but it sends a signal. And then proteins from the inside of the cell then come and wrap around that protein or the membrane that's starting to pull in a little bit. Okay? So it's starting to pull in a little bit, and you recruit these proteins that come and wrap around it. Why do they come and wrap around it? To give it structure. You remember I told you that these membranes are very fluid. Now within the whole cell, we had the cytoskeleton, we had cholesterol, we had other proteins that are sort of giving it structure. Well, these vesicles are the same thing. They don't have a lot of structure. And so we have proteins that come and sort of give it its structure, hold it together. Okay? So proteins come around and then it pinches off. And of course, these proteins are recycled, so that's an important thing to note. The proteins are recycled and used for other endocytosis, because we have to imagine this is one process, but we have millions of things going on. We talked about sodium potassium pump, we've talked about all these channels, we've talked about processes of energy production and all these things. There's millions of things going on in this one spot. Okay, so these proteins, and of course other endocytosis activities. So these proteins are recycled to go and do it again. So once you come in, the proteins leave it, and the vesicle fuses with what we've already learned as the lysosome. The vesicle fuses with the lysosome. That was an organelle that we talked about. It's packed full of enzymes. And when it fuses with the lysosome, it becomes what's called an endosome. This is a lysosome fused with a vesicle, and I want you to know that term, an endosome. And then whatever, this is, again, I told you this is very general. Whatever the cargo it is, will determine what happens after that. But for the most part, when the lysosome fuses and forms an endosome, the contents of whatever is in the vesicle will go where it needs to go after that. But the vesicle will then go back to the membrane and become part of the plasma membrane again. Because you will remember that this vesicle is plasma membrane. So what happens is it just moves up and it fuses back with the membrane. Because it is part of the membrane. However, I just want you to know these steps. Understand that depending on what the chemical is will decide whatever it does after that. But what we know to happen is that the plasma membrane invaginates, we form a protein coat leaves, the vesicle fuses with the lysosome to form an endosome, and then whatever the contents do after that is up to whatever the contents are. Okay, because there's so many. You don't know what happens after that. But we also know that that vesicle goes back to the membrane and fuses with it to become part of the cell again. The cell plasma membrane. Okay, that's endocytosis, 6 steps. Now, under endocytosis, there's 3 general categories. Okay, so we've talked, let's go back a little bit. Let me just take you all back again. We have transcytosis and vesicular trafficking. Vesicular trafficking is inside the cell, transcytosis between cells. But under that, there was a process of bringing things in, and there was a process of bringing things out. Bringing things in, there are 3 different types. We just talked about the steps. These still go through the same steps, but there's a different reason why they might come in, based on whatever the molecule is. So, the first type is called phagocytosis. This is still endocytosis, but this is endocytosis of very large things, sometimes whole cells. You remember we talked about those, I don't know if we talked, did we bring up macrophages in this class? Have I talked about them at all? Okay, well, this is typically common with immune cells, this process of phagocytosis. I do want you to know that. Oftentimes, there are cells, our immune cells, that will come and bind to bacteria. And when they bind to bacteria, will then engulf them. And through that process of engulfing, you will know that you invaginate the membrane, you form the protein coat leaves, and you fuse with the lysosome. That lysosome is packed full of enzymes that will destroy and degrade that bacteria. So this process of phagocytosis is going through the same process. This is typically very large compounds, oftentimes whole cells. Okay. Then there's a process of pinocytosis. This would be more like sodium. Okay. We're engulfing a lot of tiny particles, tiny solutes. Oftentimes, these are things that we're trying to recycle. The cell has released something off, and it could be a hormone. You release a hormone into the extracellular environment. The hormone goes and does what it needs, but then there's leftover stuff in the space, and so we will pinocytose a lot of those. It's a lot of small particles that we're trying to bring back in, usually for recycling purposes. And then there's something called receptor-mediated endocytosis. It's probably the most common one that we'll see in this course. This is probably next course. But receptor-mediated endocytosis is when some sort of molecule binds to a protein receptor on the surface of the cell. I don't know if we've talked about receptors yet, but these are proteins on the surface of the cell that are trying to send. Oh yeah, we have talked about receptors. These were the sensors. Okay, we have sensors everywhere for many different things. We've talked about them in the context of like temperature regulation, homeostatic regulation. However, those sensors are still receptors on the surface of the cell. And sometimes molecules can bind to a receptor, and then the binding of the receptor starts the process of the invagination of the membrane. Okay? So it's caused by something binding to a receptor. Now, kind of similar to phagocytosis because that process still has to occur. It has to bind to a receptor, it has to know what it's binding to. However, the difference is phagocytosis, we are engulfing large things, very large things, oftentimes whole cells. Receptor-mediated endocytosis could be, it's not going to be something as large as a cell, it could be something kind of large, but it's binding to a receptor and it's engulfing it, bringing it back into the cell. So you could say that phagocytosis is a type of receptor-mediated endocytosis. However, the difference is here, we're engulfing large particles. If it's not super large, it's not a whole cell, then it will definitely only be classified as receptor-mediated endocytosis. Okay, so you all see this, you all know these three different types and the steps that occur after each one of these things. Okay. And then there's this process called exocytosis. Now, this is the process of releasing our internal components. The cell, really, not our own, but the cells. Okay? And you would want to do this for any number of reasons. But there's two proteins I want you to know from here, and I wish this was a little bit bigger. It's two proteins. And this, of course, I've mentioned to you all this before, this is electron microscopy. This is not your typical light microscope that we've been using. This is very high definition, 100,000x. So there's two types of proteins. And they're called snare proteins. We have a V snare and we have a T snare. Okay. Cumulatively, these are called snare proteins. Okay. And each one of these letters stands for something. It's not really important that you all know, but it might be important for you all to know the V and the T. Okay. V stands for vesicular snare protein, which of course has its own thing, and then T is transporter. Okay, the snare proteins. Now there are definitely other proteins that are involved in this process, but these are the two most important. So V snares are present on the vesicle. Okay. Now these are not the same thing as the coat proteins. Those coat proteins were how we were bringing the vesicle in. We're trying to push our components out. So on the membrane itself, we have T snares, those transporter snares. On the inside, we have these V snares. Okay, or on the vesicle itself. what occurs is that the vesicle then and around, this is a simplistic drawing, but these snares are all over the vesicle, okay? All over, and we have hundreds of thousands of proteins on this tiny little vesicle doing many different things, but the snares are what's common between all vesicles to be able to get the components out. So, we have a vesicular snare. On the membrane, we have the T snare. What happens is when the vesicle gets close to the membrane, these snares will sort of lock together like this. And so you can kind of see that right now. They're sort of wrapping around each other. You all can see that sort of twisting on its edges. These are sort of linear proteins. They're string-like. Now they form alpha helices, but they're very linear. These aren't really globular, okay, like we've seen before. And so when they come together, with the help of calcium, I do want you to know calcium is involved in this process. Calcium will actually allow the binding of these two together. Then they'll sort of twist. The protein will twist. And when it's twisting, it's easier to think of it like your fingers like this, and the twisting sort of like this. And as it's twisting, the protein will then pull in opposite directions because it's still twisting. These two, you all can see that it has integral portions that are transmembrane. You all see that here? And maybe let me zoom in a little bit. Oh, I guess this is close to that again. You all know that these are inside the membrane. Well, it just so happens that the B snares and the T snares, they're proteins. These are made of amino acids, okay? Have such high affinity together that they want to completely attach. So meaning that the parts that are embedded in the membrane want to become together. Almost like Twizzlers. You all know the strings on Twizzlers are together? These things, once they become in contact with each other, will start a reaction where they start to fuse, and they're trying to push each other together. And so they're wrapping around each other. And as it's wrapping around each other, eventually it will start pulling the membrane. The two membranes together, because the vesicle is still plasma membrane, and the plasma membrane is still plasma membrane. So they're made of the same thing. As they're twisting together, it starts pulling the membrane portions of the protein together. That starts to fuse the two membranes. And it keeps twisting. And it keeps twisting until now this plasma membrane becomes part of, or this vesicle membrane becomes part of the plasma membrane. Sort of like this process. Can you all see that? It's driven by the snares. The snares have a very high affinity for each other because of chemical reactions. These things, we've been leading up to these proteins. You remember, we have DNA, RNA. RNA becomes a protein, and you know all the processes in between. Don't forget that these proteins are still the amino acids, those chemicals that have affinity for each other through positives and negatives. Okay? These two connect, and then it starts a chain reaction of events that start pulling the two proteins together, and you can see that eventually they become completely associated with each other. It starts pulling the membrane, and now as it's fusing, one thing I didn't mention is as it's fusing the membrane, it's pushing out its internal components into the extracellular space. Okay, so this exocytosis process is driven by the B snares and the T snares having high affinity for each other. B snares are present on the vesicle, T snares are present on the plasma membrane. I hope that this process makes sense to everybody. And the original binding of the two is caused by calcium. It can't happen without calcium. Calcium actually stimulates this process of these two proteins coming together and sort of locks them in the first part. And then it starts the chain reaction where these amino acids, and I'll keep saying this, these amino acids have very high affinity for those amino acids. They want to be together. And they wrap around each other and pull the membrane. And then as it's pulling the membrane, it pushes its components out. And literally that vesicle becomes part of this membrane. And as it does that, these components are pushed out. Okay?
So that's exocytosis. And so that sort of ends membrane transport, and we're going to jump into cell cycle. I don't know if we will, but we'll certainly have to talk about it now. Okay. So we'll get to what we get to, and then at the end we'll see what it's on the exam. Okay. I'm not going to put, of course, everything that we're talking about on this PowerPoint on the exam, but you will be tested on it at some point. We're very behind, and so we'll get to what we get to. However, now we're jumping into the cell cycle, and we sort of mentioned this a little bit, that cells have to divide. Cells divide from pre-existing cells, but also the most important thing that you have to do is replicate the DNA. You all know that every single cell has your entire DNA in it. Okay? And our DNA, mine and your DNA, is 99.9% similar. Right? Because all it's doing is making the protein. Well, that protein can be anything. And oftentimes it's the same thing. You have B snares, I have T snares. They're identical. You have testosterone, you have estrogen, I have testosterone, I have estrogen. Those are those are lipids, but you get the point. We have all of these things that are required for the cell to occur: cytoskeleton, microtubules. That's DNA, and it's identical between almost all of us because everybody's cell needs it. The DNA that makes us different is like 0.01% because the proteins are doing so much in the body that we don't even know. Okay? But we have to replicate the whole thing. Every single cell. And you all know that the DNA is very large. DNA is incredibly large. And so to replicate DNA is it's a very costly process. We have a lot of steps that are going on. And it requires lots and lots of energy. Okay? But it's so important that our body is okay with using that energy. That's why we haven't, developed a place where our cells have done away with energy for replacing DNA. It's because it's so important it doesn't mind to use that energy. Okay? And that's why we need food. Okay. Now, understanding DNA structure and how the cell divides and replicates is essential because it's also the starting point of many diseases, particularly cancer. Starts at the level of DNA. And this recorded lecture, I don't know if I think I might have made an extra recorded lecture to help you understand. But I think that was the PowerPoint that I did earlier. Okay. So, you all have seen this structure before. This is a different figure. But you all see this double helix. Okay? Now, you all know that this isn't protein. Why do you all know that this isn't protein? Normally when you see something like this, you think it's like a protein or something. Why do you know that this is not protein?
It's a nucleic acid.
Because it's a nucleic acid. That's another type of macromolecule. Okay? So, here you all have seen this: adenine, thymine, cytosine, guanine. You all need to know those. You all know that is a deoxyribose here. And that is a pentose sugar. 5 carbons. Then you have those phosphates that were, one phosphate was associated with one nucleic acid. Hopefully you'll remember that. And I mentioned to you that the phosphates would bind to make this blue backbone. Okay. Now you all know that these are 2 individual strands that are wrapping around each other. Okay? You already know that. And here's written out what I sort of wrote on the board yesterday, is that 3 nucleic acids make a codon. 1 codon makes a single amino acid, and multiple amino acids together through that dehydration synthesis make a protein. Okay? So you all know this structure. And you all also should be able to recognize now that between A and T we have 1 hydrogen bond represented by the dotted line, and between C and G we have 3. Okay? 3 here, 2 here. And that determines the stability of the molecule. Okay, they do, I'm not going to make you all know these things, but I think it is on your all's learning objectives. You all don't have to know the distance between each turn of DNA and the distance between base pairs. That's not important to me. That's more important for chemistry. However, so what we just looked at was the DNA double helix. That happens to be one of the most loose representations or the loose packaging of DNA. I told you that we have tons and tons of DNA. Well, we have to have levels of packing it into the body. Okay, and this is where chromosomes come in. Chromosomes are made of chromatin and chromatin, which is another name for DNA, which also has protein molecules in it. And it's not visible, DNA is not visible in the cell until we reach the mitotic phase. Okay, so mitosis, like I described to you, is only one step of the entire cell cycle, and you all will see that in a second. So it's only visible during mitosis, which you all remember was one cell becoming two identical cells. Okay, so we have to talk about the way that DNA folds. So, like I told you, that double helix is literally the loosest version of DNA that you can find in the body. And there are ways to loosen the DNA up so that you can sequence it, but it's literally the most loosest and the most unstable. Because literally all that's holding it together is hydrogen bonds. Okay. Now, there are multiple levels, and I want you to know each level of DNA. DNA double helix is the first step, so to speak, the loosest version, and it exists either as what's called euchromatin, which is open and uncoiled, or heterochromatin, which is very tightly coiled together. Okay. Now, first step is DNA, or the double helix. Double helix will start to coil together. Okay, it's getting tighter and tighter as it's going up these levels. Once it coils, it will start coiling around these proteins. Again, these proteins, that's kind of weird to think. If DNA is making the protein, why would it be wrapping around another type of protein? That just tells you the importance of this entire thing. It's wrapping around these proteins called histones. Okay, and these are globular proteins that have 4 subunits associated with them. There are 8 globules together. So you remember I showed you that quaternary structure and you all saw 2 globules together. These are 8 together, 8 subunits of a protein. They're kind of large. And DNA will wrap around them and literally create this structure called beads on a string. So histones are the proteins. They wrap around them. Now when you have multiple wrapped around, you form a nucleosome. Okay, so histones, then DNA is wrapping around histones, and the DNA wrapping around more histones becomes a nucleosome. Okay, and then the nucleosomes start to wrap around each other to become what's called a supercoil. And then those supercoils will start to wrap around each other. And that will create what's called a chromosome. Okay, so really what I need you to understand is that we have these levels that DNA exists at. Okay, and the chromosome is literally the tightest version. DNA is the loosest version. And what I really want you all to understand is that we have so much DNA that we literally have to pack it this tight in order to keep it inside a tiny nucleus. It's huge. And so condensed version is heterochromatin, uncoiled version or open version is called euchromatin. Again, whenever you see these terms, understand this is literally DNA. Euchromatin, heterochromatin, it's just talking about condensed or open state. And why would you even want to have an open state DNA? Why would it matter between these two? Now I haven't taught you all this, but again, let's think a little bit. Why would you want to have an open state of DNA?
I don't know if this is right, but would it work like the channels of like binding a protein together? Would it like bind more DNA together?
Okay, that's very close. You remember I showed you that picture of the ribosome. What did you all see there? What did you all see in that ribosome? I showed you that there was a big subunit little tiny subunit. And you know that this was RNA, but RNA has, we're talking about the same nucleic acids, with the exception of T replaced with U, still nucleic acids. What did you all see in that ribosome? You all saw a single strand of RNA. A single strand. You saw that strand cut in half. That's how we were making the protein. The RNA was being read, one strand of it, and it was adding amino acids. So why would you want to have euchromatin, uncoiled, open version of chromatin? That you would think that that'd be dangerous for DNA. It'd be exposed to a lot of stuff that might degrade it. We want it open so that we can replicate it. We cannot replicate a whole chromosome. Chromosome, 18 cannot just magically become chromosome 18 in another cell. We have to uncoil this guy so that we can start to replicate every single individual base pair, because you all know that the sequence of it has to be the same across every single cell, right? We talked about those codons, reading 3 letters at a time. Those have to be the same across every single chromosome, across every single piece of DNA. So we have to be very meticulous in how we copy this entire strand. That requires us to open this guy up and literally copy every single nucleotide, one by one by one by one by one, for every single individual chromosome. And what you all kind of already know, this is the first time you're introduced to a chromosome, we have 21 pairs. 1, 2, 3, 4, go all the way down to 48 or 46 or whatever. We have tons of DNA. And we have to replicate that guy nucleotide by nucleotide by nucleotide by nucleotide by nucleotide. It is an amazing process, and every single cell in your body does it. So this is a very important process to keep that DNA conserved. The same sequence, every single cell in your body, the billions of cells that you have to go through this process, and we just don't have one chromosome. Okay. So again, I'm introducing, we have 23 chromosomes, but I'm introducing, what's called a karyotype. A karyotype is a panel of DNA, or a panel of chromosomes. So you don't have to sequence somebody's DNA. Sometimes you can diagnose or look at somebody's, tell if somebody has a disease based on if you can see the correct chromosome parents. Okay, so oftentimes you can diagnose like Down syndrome with an extra 21 chromosome, but the point is the karyotype is literally lining up the chromosomes to see if you have every single pair that you need. Now, this is not for sequencing DNA. You cannot tell the nucleotides from individual chromosomes, okay? Because this is an incredibly packed together DNA. And so the point here is you have 23, well, most have 23 pairs, well, you do have 23 pairs of DNA. Females have 2 X, and males have an X and a Y. Okay, so you have 23 pairs. You inherit 1 from your mom and 1 from your father. Okay. Now, in the case, we'll get to the sex chromosomes shortly. In the case of females, you inherit an X from your mom and X from your dad. In the case of males, you inherit an X from your mom and a Y from your dad. And it does get a little bit more complicated after that. And we'll also talk about how the expression is with each chromosome. Now, you have to understand, that number 13 chromosome is the same as this number 13 chromosome. 1 comes from your mom, 1 comes from your dad. The magic to, what's going on here is how does the body decide which chromosome genes to express? And maybe we can think of it as a snare. You have the V snare, you have the T snare. How does your body know maybe that V snare gene is on chromosome 15? How does your body know to express your mom's or your dad's? That's a huge question in science. And it's the same right here for the X chromosome as well. You get 1 from your mom, 1 from your dad. How does your body decide which chromosome to use the DNA from? Kind of interesting. But, so you have 23 pairs. And so when I say 23 pairs, you know that you have 46 individual chromosomes, okay, of DNA. So again, the karyotype is the visual appearance of the chromosomes themselves. As simple as that. I really want to get down to